Hi everyone, this is lecture 34, Quantitative Traits and Artificial Selection. This material is found in chapter 22, and a lot of what I'm going to talk about today is going to be in page 667 to 668. So far we've talked about traits that are controlled by many genes. These are quantitative traits. What scientists want to know is how much of those traits are controlled by not just the genes but also by environment. This has broad implications. For example, in agriculture, when artificial breeding experiments are conducted, you want to know how effective will breeding programs be if, in fact, the environment is what's controlling the trait. So let's see how this works. Question number 1. In an artificial breeding experiment, a population will likely respond to selection if it exhibits what? Number 2, kind of a long one. 2 true breeding tobacco strains were crossed, one with long corollas, which are flowers, and one with short. The F1 generation resulted in a distribution of corolla lengths, with an average of 50 millimeters. Next, an F1 self-cross was performed, with a plant having corollas 42 millimeters long. The resulting F2 generation had a distribution in length, with an average of 46 millimeters. What is the narrow sense heritability of corolla length in this tobacco strain? So, when we're talking about traits that are controlled not just by the genetics but also by the environment, we call these multifactorial traits because there's multiple factors controlling them. The genetic portion are often polygenic, think quantitative, meaning multiple genes are affecting the trait. And the environment portion is this old argument of nature versus nurture. How much of the trait is controlled by the environment? We're going to define this term phenotypic variance, which is the variation in a population. In this picture, the phenotypic variation is graphically displayed, whatever the low versus to the high, whatever it is you're measuring. This phenotypic variance is a combination of two components primarily, the genetic variance and the environmental variance. Though from looking at this, it's impossible to determine which and how much of this is controlled by each. The environment serves to blur distinctions between discrete phenotypes. You might recognize these normal distributions, these bar charts, as what we looked at with the wheat color, the red versus white wheat, where there were certain categories even though they resembled a normal distribution. The environment, when overlaid on the genetics, tends to blur the lines because it makes each phenotype either perform better or worse. And so you get a gradual range in phenotypes as opposed to discrete phenotypes. To illustrate how genetics and environment can control a single trait, let's look at this example from your textbook. In the upper left-hand corner is a sample plot which shows two different strains of plants, which are labeled G1 and G2 for genotype 1 and genotype 2. These two plants were grown at different temperatures from 15, 20, 25, and 30 degrees Celsius, much like you might do in a middle school science fair project. After some time, their height was recorded and it's graphed. I think what you can see from panel A is that the height in the plants is a factor of the genetics. The plant called G2 has done better than plant G1. That means the genotype of plant G2 is outperforming the genotype of plant G1. The temperature makes no difference at all. Compare that to panel B, where now temperature clearly makes a difference in how well they grow. Simply just growing at a higher temperature makes both plants grow better. Both G1 and G2 are overlapped in this picture, that's why they show up green. Compare that to panel C down here at the bottom, where both genes and environment are making an impact. Clearly higher temperature is making them grow better, but still G2s are growing better than G1s. So it's a combination, both genetics and environment. And then finally, in panel D is a more complicated example where another factor called gene by environment interaction is taking place. The environment definitely makes a difference, but it's having a differential effect on the two different plants. G1 plants are growing better with increased temperature, whereas G2 plants are growing worse. We're going to be using artificial selection as a tool to understand how do populations change and how rapidly can they change in response to genetic breeding experiments. It gets us to the point of how much of a trait is controlled by the environment and how much is controlled by genetics. So artificial breeding or artificial selection is the process of choosing specific individuals with preferred phenotypes from an initially variable population for future breeding purposes. And here's the important part. The degree of the success depends on the relative contributions of genetic and environment. If a trait is controlled mainly by genetics, artificial breeding will be very successful. On the other hand, if the trait is controlled by the environment, well, as we saw with the plant example, just simply growing the plants at a higher temperature makes them do better. Artificial breeding is not going to do so well for you there. Let's look at some examples. Here's a population that shows phenotypic variance. We define a term called S, which is the selection differential when we do artificial selection. S is the difference in the phenotype of an individual from the population, from the mean of that population. In this picture, the dashed line is representing the mean population. The shaded blue area in this part of the graph is the region from which an individual is selected. This could be plant height. Let's pretend it's plant height. So selecting a tall plant from this population and breeding those two tall plants together would expect to give you more tall plants in the next generation. And in fact, that's what you see. Here, the next generation has an average height that was the original average height of the parents. We define this term R, or the response to selection, as the change in phenotype from the original population mean. Here, in this case, as you can see, S is equal to R. When S is equal to R, this happens when the variance that we see, the phenotypic variance, is determined solely by the genetic variance. That is, additive alleles. Some alleles are adding to the trait, and others are not. I want to define a term now called heritability. This value can be estimated by how rapidly a population responds to selection. If a trait is controlled primarily by the genetics, then it will respond rapidly to artificial breeding experiments. If a trait is controlled mainly by the environment, it will not. Heritability helps us figure that out. Heritability then is equal to the response to selection divided by the selection. Another way to write that in a formula, heritability is given the symbol HN2, and it's equal to R divided by S. In this example, where R is equal to S, we can say that heritability is equal to 1. A heritability value of 1 means that the variance seen in the population is solely determined by genetic variance. Artificial breeding experiments will be very successful. On the other hand, here's a population that when two tall plants were selected, again, the plants on the right-hand side of the graph, and they were crossed, the next generation showed no change in the height of the plants. In fact, the next generation was exactly the same as the original parents. This happens when the phenotypic variance seen here is determined solely by environmental variance. Here, R is equal to 0, and so heritability will equal 0. So, heritability can range between a value of 0 and 1. A trait with heritability of 0 indicates that all of the variance is due to environment. Artificial selection will not help you. Just simply grow the plants at a better temperature or give them more water, better nutrients. As opposed to a heritability of 1, which indicates that all the variance is due to genetics. Breed the tall plants together and you'll get more tall plants. The reality is, though, it's more complicated. And here's an example where the phenotypic variance is seen in the population, 2 tall plants are selected, and the next generation is a slight increase in height, but it's not what you would expect with a complete a heritability of 1. So from this you can calculate what the heritability is by dividing R divided by S, and those are the kind of problems we'll work in class. All right, so see you then.